Hello, everyone. Just wanted to get us up and started. I know it's the top of the hour, so I'll actually just get us going. So again, hello, and welcome to Fight Colorectal Cancer's very first clinical trials webinar. I'm Dr. Gitanalagan, part of Fight CRC's research advocacy team. For those of you that may not be familiar with Fight CRC, we are the leading patient empowerment and advocacy organization in the United States, providing balanced and objective information on colon and rectal cancer research, treatment, and policy. We are relentless champions of hope, focused on funding promising high-impact research endeavors while equipping advocates to influence legislation and policy for collective good. Again, on behalf of Fight CRC, we are so happy to have you with us for the launch of this new series. Our goal with these webinars is to bridge science, advocacy, and patient empowerment, helping our community better understand clinical trials and how science-driven research impacts real lives. Before we begin, a few quick housekeeping items. We will have some dedicated time towards the end of the webinar for general questions, but we encourage you to please use the Q&A panel on your screen at any time during the event. This webinar is meant to educate as well as serve as a forum for you to ask your questions and start conversation. So please don't be shy. We will do our best to answer any and all questions that are asked, but do please keep in mind that we may not be able to answer any specific questions about your treatment or diagnosis during this event. We will have the recording of this webinar available on our site in the next few days, and you will receive that direct link via email as soon as it's available if you registered for this webinar. We also will provide a transcript of this webinar on our site for those of you that would prefer to read the information discussed. Also, feel free to tweet along with us. You can use the hashtag CRC webinar. To get into some of our resources, please remember to stop by our website at fightcrc.org to check out all of our patient and caregiver resources, such as our clinical trials newsletter, where you can subscribe to receive research updates directly to your inboxes, and it'll allow you to kind of keep in the know for any of our future webinars on clinical trials. We also offer a variety of free-to-use online tools, such as the Patient Provider Finder, which allows you to search for providers in your area with experience and expertise in treating CRC. There's also a free Community of Champions app where you can connect with other people in the colorectal cancer space and keep in touch with Fight CRC to know what's going on. And there's an assortment of print and digital educational resources that are free to request and download on the site. Just as a quick disclaimer, the information and services provided by Fight Colorectal Cancer are for general informational purposes only. The information and services are not intended to be substitutes for professional medical advice, diagnoses, or treatment. If you are ill or suspect that you are ill, see a doctor immediately. In an emergency, call 911 or go to your nearest emergency room. Fight Colorectal Cancer never recommends or endorses any specific physicians, products, or treatments for any condition. Now, to meet our amazing panel of speakers, we are honored to welcome three incredible experts and advocates. First, Dr. Al Benson, Professor of Medicine at Northwestern University and Director for Cooperative Groups at the Lurie Cancer Center, who also serves as Fight CRC's on Fight CRC's Medical Advisory Board. Dr. Benson has been instrumental in advancing GI cancer clinical trials and biomarker-driven treatment guidelines. Also with us is Dr. John Strickler, Professor of Medicine at Duke University and co-leader of the Precision Cancer Medicine and Investigational Therapeutics program. Dr. Strickler led the MOUNTAINEER clinical trial, which paved the way for the first FDA-approved therapy for HER2-positive metastatic colorectal cancer. And last but not leastly, we have Daad Abighanem, a stage 4 colorectal cancer survivor and Fight CRC dual role research advocate and ambassador. Daad's experience and advocacy bring the essential patient voice to research and policy that grounds today's discussion. So we will begin now with Dr. Benson, who will walk us through what HER2 means in colorectal cancer and how testing guides treatment.
Oops, you're muted.
There. Sorry, can you hear me? Thanks so much, Karen, for this opportunity to join today's discussion. You just heard the words precision medicine, and this has really been a phenomenal development, which has enabled us to not only better understand the biology of tumors and what makes them grow and spread, but it has led to the development of very targeted treatments, which have certainly helped our patients. So today's discussion is a particular targeted approach, representative of precision medicine, referred to as the HER2 gene that stands for the human epidural growth factor receptor 2. And this is also often referred to as a biomarker. And so you'll often hear people talking about the importance of biomarker testing. What you see on the right is actually a simplified diagram of the very complex interaction of various biological pathways within the cell that can lead to growth of tumors. And HER2 is part of the HER2 and EGFR receptor family. And you can see the various family members listed here. The HER2, or also often referred to as the ERBB2 gene, is in each of the cells in the body and is in the same family as the EGFR gene, which is also another target in terms of cancer therapies. The HER2 protein is also a receptor, and I'll show you a diagram in a minute. But this gene can provide instructions for making a protein that is involved in cell growth and division. Next slide. Now, the HER2 protein, as I mentioned, is a receptor, which is like a microscopic spike that sticks out on the surface of almost all of the cells in our body. However, these HER2 genes are responsible for communication between cells to promote their growth, division, and repair, as well as survival. Now, a normal cell and its nucleus generally have just a few HER2 receptors, but sometimes with abnormalities within the cell, multiple HER2 genes are actually produced, and this is referred to as amplification. When the HER2 gene is amplified, it can produce excessive numbers of these receptors on the cell surface, and this is referred to as overexpression. And this overexpression can drive this uncontrolled growth of cells, which become tumors, which can eventually spread. HER2 amplification is actually relatively uncommon. It's estimated to be present in 3 to 5% of metastatic colorectal cancer patients. However, it is more common for patients who have what's referred to as wild-type KRAS. And KRAS testing is a routine aspect of precision medicine and what we routinely evaluate for our patients, particularly when they have metastatic disease, because it too can influence the type of treatment we might choose for an individual. Also, what's important is that this amplification can lead to resistance to some of these targeted EGFR therapies. Next slide. Now, it's important to identify whether an individual has HER2 overexpression or amplification. And this testing is done by our pathologists. And it is important because it can help identify patients who may benefit from specific targeted therapies. In fact, HER2 testing now is recommended for all metastatic colorectal cancer patients, and this is included in cancer guidelines, including the guidelines of the National Comprehensive Cancer Network. Often, our testing is more comprehensive through NGS, which stands for Next Generation Sequencing. The presence of HER2 overexpression is most typically seen for those individuals who have RAS or BRAF wild type tumors, and also those with microsatellite stable tumors. And these also represent markers that we routinely evaluate for our patients. There are some common testing methods. One of the most common is immunohistochemistry, and you can see what that looks like on the right side of the slide. So what the pathologist is looking for is the presence or intensity of staining, which is directly looking for HER2 expression. And when the pathologist is evaluating these stained pathology slides, there is a score which ranges from 0 to 3 plus. And so you can see the difference here. Those are the top slides that go from 0 to 3 plus. And the individuals who have 3 plus staining or overexpressing tumors are those that are most likely to benefit from a HER2-directed therapy. What you see, the bottom 3 slides represent what's known as in situ hybridization, which demonstrates gene amplification. And that shows progressive amplification in those bottom 3 slides. I did mention NGS, so that's another way that we can detect the HER2 status as well as other important markers. So what we look for are individuals who are positive for HER2 overexpression or amplification, and that's defined as those who have the immunohistochemistry 3 plus score or a score of 2 plus, but also show the in situ hybridization amplification. Next slide. So why does this matter? As I mentioned, it can identify those who may benefit from targeted therapy, and Dr. Strickler is going to talk a great deal about this in a few minutes. Early identification can also prevent exposure to agents that may be ineffective, such as those agents that target EGFR. It also is very important because HER2 is still a very important area of research. And so we continue to do multiple clinical trials exploring treatment opportunities for people who have HER2 overexpressing tumors. It also helps inform treatment decisions, and importantly, how we might sequence treatments over time to best benefit our patients. And more, this precision oncology planning is important because we are developing more and more treatments for specific biological targets that we can identify in tumor samples. So the bottom pathway shows you that we can take a tumor, do HER2 testing, identify if it's positive, identify a targeted therapy, and even the possibility of a clinical trial. Next. So we have, fortunately now, multiple therapies that target HER2. You're going to hear from Dr. Strickler, who will provide much more detail about these targeted therapies, but here are some examples of Ducatinib plus trastuzumab, the MOUNTAINEER study, but also trastuzumab can be combined with other agents which have led to benefit for our patients as listed here. And also more recently, the trastuzumab deruxtecan agent, which it also expands our ability to help people with HER2 positive tumors. So these regimens are available options following initial therapy for patients who have HER2 positive RAS BRAF wild type metastatic colorectal cancer. Next. So I mentioned we continue to conduct clinical trials. So there are emerging therapies and combinations, just some examples listed here, including next generation tyrosine kinase inhibitors, as well as this growing area of drug development referred to as antibody drug conjugates and these dual targeting approaches to help our patients. And so there's a research focus to also optimize sequencing and combinations with chemotherapy and immunotherapy, as well as using HER2-directed therapy earlier in the treatment landscape. Next. So in terms of future directions, there's clearly an effort now that we've shown that HER2-targeted therapy can help people who have already been exposed to a previous regimen to see how well it works as perhaps the first therapy for someone with metastatic disease. There's also a great deal of focus on resistance mechanisms because unfortunately, although our targeted therapies can often be very helpful and control disease for a while, eventually resistance sets in and people no longer benefit and need to have an alternative approach. As mentioned, we're also looking at novel combinations, including combinations with immunotherapy. It's very clear that although it's a relatively infrequent event, it's still an essential biomarker for precision treatment in colorectal cancer. And with our growing number of clinical trials and the help from advocacy groups, we continue to make progress in this particular area of HER2-directed therapy. And I think that's my last slide, and thanks for your attention.
Thank you, Dr. Benson. You've really set up a strong foundation for helping us understand the science behind HER2 and colorectal cancer. Before we move on to Dr. Strickler, I'd like to take a moment to address a couple of questions that were submitted ahead of time before today's webinar. And these are some of the most common questions that we hear from patients and caregivers. You've touched on them, but if you could really just drive home these 2 points. First being, why is HER2 testing important for all patients with metastatic colorectal cancer? And then secondly, if you could explain to us how does HER2 testing in colorectal cancer differ from how it's used in other cancers like breast or gastric cancer?
Yes, so we used to treat colon cancer as if it was just one disease and everybody receiving the same therapy. And in the past, we were actually quite limited in the agents we had, and they were principally chemotherapy drugs. But through the advances of technology, we've been able to realize that colon tumors differ across our patient population. And it became increasingly obvious that identifying subsets of patients who had particular molecular characteristics demonstrated in their tumors were such that not only could it affect outcomes, but also provided the opportunity to actually develop treatments specific for those biological parameters. And so certainly over the past 10 years, we've made significant advances, and certainly with the utilization of next generation sequencing, or NGS, we're able to identify quite a broad array of different markers to determine if for an individual patient there's a particular molecular variant for which we have a therapy. And understanding this is really critical as we think of what would be the best treatment opportunities for an individual over time. And often people with metastatic colorectal cancer require multiple different treatments over time. And by doing so, we've certainly been able to have our individuals live better quality of life and to enjoy longer life. And so we urge this testing at the time of diagnosis so we can better plan a treatment strategy. Although HER2 overexpression is seen in other tumors, such as stomach cancer tumors, and certainly in breast cancer tumors, the incidence of that expression varies across these diseases. And in some cases, the treatments are very similar. Not always, but very similar. There are other diseases where this testing is pretty essential. But for these diseases, different treatment parameters are utilized in terms of the expression.
Thank you, Dr. Benson. I know that there's a couple of questions that are coming in already, so we'll hold off until open Q&A later in the webinar. So right now we will move on to Dr. Strickler, who will walk us through the MOUNTAINEER trial and discuss what's next in HER2 targeted research and treatment.
Oh, thank you so much. Pleasure to be here today. I appreciate the invitation. I'm John Strickler from Duke University. And here are my disclosures. So let's start with some key top-line messages from what I'm about to cover here. The first is that MOUNTAINEER was an international phase 2 trial that looked at tucatinib trastuzumab and tucatinib by itself in patients with HER2-positive RAS wild-type metastatic colorectal cancer. This study showed that tucatinib and trastuzumab was generally well tolerated by most patients, and most importantly, had clinically meaningful activity. And based on that, it was FDA approved. And I think as a core part of our anti-HER2 treatment algorithm for patients with metastatic colorectal cancer, and I'll show you some data to kind of follow that point here. Now, I'm involved in precision cancer medicine, and that's my mission as a clinical investigator, to bring these therapies into the clinic. And when we talk about precision cancer medicine, what we're really saying is we want to bring the right treatment to the right patient at the right time to give that patient a treatment that's both well tolerated and active, sparing that patient treatments that are either toxic or ineffective. And MOUNTAINEER is really a textbook case of precision cancer medicine. So at the time that I developed this trial, because it was a trial that I wrote myself, I was finding HER2 amplification in my patients with colon cancer, but I had no treatment for it. And many of these patients were heavily pretreated, had exhausted FOLFOX, FOLFIRI, these chemotherapies that can be very hard on the bone marrow and quality of life. And I was looking for the cleanest, most active treatment that gave people a good quality of life. So tucatinib was my first choice when I was surveying the landscape of treatments, because if you can see in this, I don't know if I have a pointer here, but as you can see here on the right, there's a low number for HER2 IC50. That means it's very potent against HER2. And if you look here to the right, this EGFR high number of 450, that means it's not potent for that target. And when you attack HER2 exclusively and spare EGFR, you get a lot better tolerated treatments, less rash, less nausea, less diarrhea. And that was the inspiration behind this combination. What could we give patients that makes a well-tolerated, highly active treatment without a single dose of chemo? And this was the, originally I designed this as cohort A, if you put the pointer around cohort A, and it was meant as a pilot. We're going to treat 25 patients with tucatinib and trastuzumab, and who have HER2 positive RAS wild type metastatic colorectal cancer. All the patients have received prior standard chemotherapy, and just see how it goes. And the initial impact was so significant that the company I was working with said, gosh, we want to make this more widely available to patients nationally. So let's expand this trial, make sure we know that it needs to be tucatinib and trastuzumab, not tucatinib, and potentially go to the FDA for an approval. So that was the inspiration behind this design. It was meant to be really by the doctors for the patients kind of trial. And here were the patients we enrolled to cohorts A and B. These are the patients that got the tucatinib trastuzumab combo. We also looked at tucatinib by itself. The things I'll point out here, I've put a box around this. It makes a point about where you find HER2 positive RAS wild type colon cancer. It typically is on the left side of the colon or the rectum. So if you hear things like the descending colon or the sigmoid colon or the rectum, that's really the bulk of these cases. The other thing that's interesting is these patients are more likely to have lung metastases than a standard colon cancer patient. So some things to be aware of that we found just in enrolling these patients. Additionally, sorry, I'm advancing to the next slide. Summary on the summary of prior anti-cancer therapy slide, many of these patients had very heavily pretreated disease, so they had more than 3 lines of therapy. So most of the patients had received 5-FU, oxaliplatin, irinotecan. It just makes that point that these patients had received a lot at the time they were enrolled. And this is a setting where the FDA-approved standard of care generally generates low response rates and controls the disease for a short amount of time. So advancing to the next slide, looking at the safety summary here. And the thing I will point out is the goal of this was to make it clean, meaning as few side effects as possible. And in fact, that's what we found. There were very few patients who had a serious or a life-threatening adverse event. And it was very rare for tucatinib to be held or dose reduced due to a side effect. And so that was a goal, and that was a success out of this trial. And then advancing to the next slide, you can see the most common adverse events. You can see very little orange. Those are the serious side effects. It's mainly blue. So grades 1 and 2, the tucatinib trastuzumab combo most commonly will cause diarrhea, but in most cases it's controlled with things like Imodium, like standard over-the-counter medicines. In my experience, the most serious adverse event that would lead to a dose reduction would be liver enzyme abnormalities. And those are pretty easy to catch, and it will resolve in most cases pretty soon after holding the drug. So how well did this combination work? Let's move to this next slide. On our left, I'm going to focus on tucatinib and trastuzumab because that's what ultimately became FDA approved. In a setting where the response rate for the FDA approved therapies is usually less than 5%, we were able to generate a response rate of over 39%. And what it was even more impressive is how long patients benefited. So when we generate a response, typically that response would last well over a year, sometimes 2 years. In fact, we have a patient we enrolled who had a complete response that remains to this day over 5 years with very few side effects. So, some real victories here. But the average person stayed on this regimen for 8 months, over 8 months, which when you consider the alternatives in this setting looked really great, particularly given the quality of life. And in fact, when I would enroll patients on study and the treatment stopped working, the patients sometimes would ask me if they could still stay on anyway, because it was the regimen they had tolerated the best at that point for their disease. And in fact, that's what we do a lot now is when a patient has progression, we will either remove or radiate that progressing lesion and keep the person on tucatinib and trastuzumab because the quality of life is so good in most cases. So advancing to the next slide, as I had mentioned, some patients experience really deep and durable responses, and this what we call spaghetti plot on the right really drives home that point. All the blue lines are people who responded, and you can see there's a sizable group of patients who are blue, where that response goes on for literally more than 3 years. Not everybody, but it has been a success for a sizable group of patients. And it's a real win when you can put a patient into a durable response with a regimen that's well tolerated. So that was really a heartwarming for me to see this curve. I think that 5 patients out of 60 had a complete response where there was literally no cancer left seen on scans. So it's not common, but it does happen. Moving on to this next slide, this is starting to get into some technical matters. I think Dr. Benson had mentioned this idea of IHC, which is really our gold standard for diagnosing HER2 positive disease. I think the key point to take away here is that if you look at this table on the right, where it's IHC 3+, that's where we see the best responses to this regimen with a 46% response rate. It does drop off the lower the level of expression. So 3+ is high, 2+ is medium, and negative is basically 0. And you can see the response rate really drops off as the expression goes down. On the left, just wanted to make the point that some patients get anti-EGFR therapy, some people didn't. It didn't seem to impact response to this regimen. Advancing to this next slide, I'm going to skip over this for the interest of time, but the bottom line is we can count the genes on the chromosome, and the higher the HER2 copy number, the more you benefit. That's kind of making a long story short here. So we'll advance to the next slide. We also do a lot of circulating tumor DNA in our clinic to look at whether other mutations and alterations can drive responsiveness. And interestingly, it didn't seem like these other mutations and alterations in blood really had an impact on how well somebody responded. So while this is approved for patients with RAS wild type disease, there were some patients enrolled who had RAS mutations detected in blood, and they did not seem to drive resistance to this regimen. So it's kind of an interesting, interesting point from this. So as we advance to this next slide, patients in the clinic we're getting the molecular testing, but there's all kinds of different ways we're doing it. And this slide is really important because what this is telling us is in this first column, looking at tissue IHC in FISH, which is kind of our gold standard for testing HER2. Here we can see a response rate of 41% among those patients who are positive with a long progression-free survival. But look in this next category, tissue NGS. Most patients are getting NGS test and not IHC. Here, the response rate's similar at 50% with a nearly identical progression-free survival. There are some people, though, that can't get tissue testing for whatever reason, that's this last category of blood NGS. And we can see here, if you're positive by a blood NGS, similar response rate at 42% with a progression-free survival at 8 months. The one thing I will say is you can use a blood NGS test. Some people call those Guardant tests or whatever. There's different vendors. But you can use it to rule in HER2 positive disease. You can't use it to rule out HER2 positive disease. So if it's not detected in blood, it doesn't mean you don't have HER2. It means you really got to go back to tissue to figure out if you have it or not. Because there were some patients who had no HER2 positivity detected in blood who still responded. And that's because the HER2 wasn't detected. It was there, it just wasn't seen. So that's a blood issue that you need to be aware of as a patient or family member. And then finally, we've also learned a lot about resistance. Unfortunately, most of the patients enrolled eventually did develop progressive disease. And this is a blood test that we did at the end of the trial for these patients who progressed. Each row is a patient. And what you'll see is lots of different colors light up. And these colors represent different types of genetic mutations or alterations. And what we found is that in the majority of cases, the reason why the cancer progressed on tucatinib and trastuzumab was because of one of these acquired alterations, and that drives resistance. Interestingly, very few patients lost HER2 at the time of progression. This is really important because it's going to set up these next slides here. So if we advance to this next slide, there are other therapies in the clinic. One of them that's FDA approved now for patients with IHC3+ disease is a drug called trastuzumab deruxtecan. This was tested in both the Destiny CRC01 and 02 trials. What I'm going to focus on here is cohort A from this trial, where they looked at the response rate of this drug. And if we advance to this next slide, this so-called efficacy outcome slide on next, we can see here that the response rate for this drug, it's called an antibody drug conjugate, was 45%, duration of response at 7 months, progression-free survival at 6.9 months, which looks fine. However, if you advance to the next slide, there was a problem with this initial trial, which is that many patients experienced serious lung toxicity with this therapy. In fact, there were 3 deaths out of the first 86 patients enrolled, and that's obviously unacceptable safety. So the question was, well, maybe we need to get the dose right. So if you advance to the next slide, that's the logic and the inspiration behind this Destiny CRC02 trial, where they tested a lower dose of this drug compared to a higher dose. And what they found on the next slide is that at the lower dose of this antibody drug conjugate, you get a higher response rate, similar progression-free survival, and overall more favorable outcomes. And if you advance to the next slide, you can see here that there were no deaths from this interstitial lung disease at the lower dose. So it's not, more is not always better. With this drug, it's an antibody drug conjugate, less is more. Take the 5.4 milligram per kilogram dose, and that's actually what the approved dose is for this now. If you advance to the next slide, just some really important messages. Number one, most patients who benefit from this antibody drug conjugate have the highest level of expression, IHC3+. This treatment benefits people whether or not they have a RAS mutation, and most importantly, at the bottom, it does not matter if you've progressed on tucatinib trastuzumab, your response rate is the same. So that's why this drug almost always should follow after tucatinib trastuzumab, because this can salvage a response and generate a response even if somebody progressed. The contrary is not true. I would not give tucatinib trastuzumab after trastuzumab deruxtecan. We've never shown that that's an effective strategy. So if you look at this next slide, this is the therapeutic landscape for HER2 positive metastatic colorectal cancer. Key thing to know here is you want to be up here in the top right-hand corner with the highest response rate and the highest progression-free survival. And so that's where you have your FDA-approved therapies. And that's, if you advance to the next slide, these are FDA-approved therapies shown here, and approved in 2023 and 24. And then finally, wrapping up here on this last slide, how I treat HER2 positive metastatic colorectal cancer. If there's no RAS mutation, I would give tucatinib trastuzumab after a round of chemotherapy, and then use trastuzumab deruxtecan to salvage a response if the patient develops progression. And so we are not done making breakthroughs for HER2 positive disease. This next slide shows our MOUNTAINEER 03 trial, our first-line trial, where we're putting tucatinib and trastuzumab together with FOLFOX to see if we can bring these drugs earlier into the clinic for patients. So hopefully, we will fill enrollment to that soon and report that out. I'm cautiously optimistic that it will come in the future. And then finally, there are a number of different therapeutic strategies in the clinic to target HER2. We've got targeted therapeutics like tucatinib, immune therapies, antibody drug conjugates, and radioligands. All of these are exciting therapies to help make breakthroughs for this disease. So where are we going with HER2 for future directions on the next slide? Oops, sorry. I think bottom line, tucatinib and trastuzumab is generally safe and well tolerated and should be a standard option. And we use trastuzumab deruxtecan in cases of progression, but we are aware of this resistance issue. So we are developing new therapeutic strategies to overcome that. Sorry for going a little bit long.
No, not at all. Thank you so much, Dr. Strickler. This definitely helped us better understand how MOUNTAINEER study has changed really what's possible for HER2 positive CRC. Before we move on, I did just want to see if you could answer one question that is just, I think, hits on a lot of patients that wonder, how can patients and advocates stay updated on new and or ongoing HER2 related trials?
Not an easy question to answer because the doctors barely know what's going on. It's a very rapidly changing landscape. I'm not just saying this because I'm on the Fight CRC webinar right now. I actually encourage my patients to reach out to patient advocacy organizations. Patients look out for one another and are aware of things nationally. Doctors have a tendency to get a little bit tunnel vision around what's going on at their own institution. So I would say definitely use the resources from Fight CRC, from your patient advocacy organizations, because these are the groups that can look well outside a doctor's institution of what's going on nationally. And of course, Al Benson's on our NCCN guidelines committee. So when something makes it and shows efficacy, then Dr. Benson will update the guidelines with his committee and make sure that those are also widely available to doctors so they're aware of those breakthroughs.
Thank you so much, Dr. Strickler, for your kind words and honestly for kind of setting up the stage perfectly for Daad, who now next will be talking to us from the patient advocacy perspective, who uses both her scientific background and lived experience to educate others and champion biomarker awareness.
Hi, everyone. Thank you for having me. Thank you for being here. It's a pleasure to meet all of you. And thank you, Dr. Benson and Dr. Strickler, for this amazing introduction to HER2 therapies. A little bit about myself and my journey with colorectal cancer. So my name is Daad Abighanem. I am a sister, a daughter, a wife, a cat mom, a dog mom, a scientist, and a stage 4 rectal cancer survivor. I was diagnosed in 2022. I had just turned 48 when I diligently went in for my annual physical. And my doctor told me, well, you're now eligible for colon cancer screening because the age of screening was lowered to 45, just, I believe, the year before, sometime in late 2021. So I did, long story short, I went in for a colonoscopy and woke up to words that were unknown to me, adenocarcinoma and, mass, rectosigmoid mass, 4 centimeter rectosigmoid mass. And what followed was a whirlwind of, how it goes, doctor appointments, tests, scans, um, first opinion, second opinion, third opinions, and a whole new barrage of information and new terminologies, some of which I was familiar with because of my science background. I was, at the time, I was working at a biotech company. I was the research and development director for a biotech company, and we basically, our work was developing antibodies and immunoassays. So that background helped me a little bit understand what's going on, but there's so much more I didn't know, and I took it upon myself to educate myself because I wanted to know exactly what's happening. I wanted to know where my tumor was, and all those terminologies and my pathology report, what did they mean? Because I early on, and I think I didn't know that at the time, but I know now that early on I wanted to be equipped to be able to ask questions to my medical team. I wanted to be part of my medical team. I didn't want just to be um, um, an innocent bystander that just follows what the doctor says. My treatment was I had surgery and then I had positive lymph nodes, so I had to have chemotherapy. I did 12 rounds of FOLFOX, and midway, I think, through my treatment, we discovered that I had a single metastasis to my lungs, to my right lung, and that required a microwave ablation of that nodule, which came back, and then we repeated the ablation again. And as of, I want to say April, I have been NED, no evidence of disease, for a year and a half. So I actually had a scan early this week, and I'm still waiting for the results. So fingers crossed. While I was during chemotherapy is when I was introduced to fight CRC. I was so lost, and so, I mean, anybody can know that a cancer diagnosis is so isolating. And I was looking for support, which I found in the fight CRC community with people who very generously offered their time and their advice, and that just basically listened to you. And that brought home to me, after I was done with my treatment, what I want to do now with what has been given to me is I want to carry this torch forward, and I want to help other people. So I joined the Fight CRC advocacy program exactly because I want to raise awareness about everything that we've been talking about today: biomarker testing, clinical trial participation. I want survivors and to be aware of what's going on in the research side. I want to empower them to ask questions, to advocate for themselves, because they need to be a part of their medical team, and they need to be given a seat, a permanent seat at that decision making table. Next slide, please. We talk about biomarker testing a lot, and early on, I learned about those biomarkers when I read my pathology report. I had to figure out what was MMR and KRAS and BRAF and all those names. Later on, I was introduced to other names like HER2 and ctDNA and tumor burden. And I want everybody to understand, I want every patient to know, every diagnosed patient to know what these mean. What do they mean for their personal care plan? I want to empower patients to ask about these biomarkers because biomarker testing empowers you. If you know your biomarkers, you know what questions to ask about your surveillance, about your treatment option, and that allows you to participate in your own care. And as as Dr. Strickler mentioned, that a lot of times doctors don't know what's going on, but knowing your markers and being connected to a community of researchers and advocates connects you to research projects, and you know that sometime in the future, if that were to happen, if you needed an alternative treatment, what could what options would you have? And I remember having that conversation with my oncologist when I did do they did do a, my tumor mutations report. I was discussing it with her, and I think she was trying to comfort me, and she was like, she said, don't worry about it now, we're not there yet. And I wanted to say, and I told her, no, we are there yet, because I need to know what happens if chemo doesn't work. Like, I need to know, is there something else we can try? And that was because I had a mutation. I have the KRAS G12C mutation. So she told me, yes, there is an option for you if we get to that. So that gave me some mental peace, some mental peace of mind. Next slide, please. So from my own experience and from talking to people, to patients, to advocates, I know there's a lot of barriers to biomarker patients, and a lot of among some of these barriers among patients and among providers as well is the limited awareness of why biomarker testing is important, and why is it important to know what your markers are, and the importance of knowing that a biomarker has a role in your treatment options. I would say a big barrier is cost. Insurance coverage. Insurance always is a problem. And I know I have heard personal stories where patients would say, no, I don't want to do this because I can't afford to pay for this test. And we also have barriers that stem from disparities depending on where you live, or your income status, your socioeconomic status, your age, your education level. It's been reported that patients treated in community settings or in rural settings versus big academic or big hospitals are less likely to receive this biomarker testing. Another barrier that I hear talked about is the evolving biomarker landscape, that there's always also research being done, and patients and clinicians are usually don't always keep up with the evidence or with what's going on. And for clinicians, it's sometimes very challenging if there's new guidelines or new evidence to integrate it into their clinical workflow. Next slide, please. Similar barriers are present that in clinical trials that prevent patients from participating in clinical trials. And we know, and there's no harm in repeating that clinical trials are the backbone of cancer clinical research, and that's how we get new treatments. And but I read a statistics that I thought was insane, that only 3 to 5% of eligible adult cancer patients actually participate in clinical trials, which obviously leads to delays in the advancement of research. And the cause for this low enrollment is a wide range of factors that hinder participation, and these are factors that are sometimes complex, multifaceted, and they can build on each other or reinforce each other. So I'll just go over some of these barriers. At the top of them, at the top of the list is the mistrust of experimental procedure, a mistrust of science, especially among underrepresented communities. And that mistrust stems from historical abuses and from systemic inequities. Patients are afraid to be treated like experimental subjects, especially when you tell them it's a clinical trial. I mean, it's in the world. They think that you are trying just something on them. And they want reassurance that the care offered in the trial is not inferior to the standard of care. And I think clinicians fail to provide this assurance. Other barriers are, again, health insurance coverage, who's gonna pay, and related to that, and the financial and logistical burden. Participants in clinical trials often have to travel long distances to the center where they're getting their treatment. They have to take time off work, find childcare, and sometimes that becomes prohibitive. I personally did not participate in a clinical trial, but I know from experience, when I was diagnosed, I was living in upstate New York in a rural area, and but I opted to, because of some barriers regarding treatment and regarding biomarker testing and all of that, I decided to go to Memorial Sloan Kettering for treatment. So every chemotherapy appointment that I had to go to, I would have my husband would drive me 3 hours to New Jersey back and forth. Every time I wanted to see my oncologist, we had to drive 4.5 hours to New York City and stay in New York City. And the hotel rates in New York City. I am very fortunate and lucky that I was able to do that. And I know a lot of people, this would become very prohibitive, something like that. Other barriers to clinical participation that I hear being talked about is a language barrier. It's understanding those extra long consent forms, the medical terminology. Another factor is some among immigrant communities, an immigration status can be a barrier because some patients are afraid that they're going to become part of a database sometime somewhere, and that will affect their immigration status. Last barrier I want to talk about is lacking support for care partners. And I think this is across care, not just clinical trial barriers. We don't think enough about caregivers who play a big part in the patient care. For example, if a person participating in a clinical trial has to go to a hospital and stay for 3 days for their treatment, can their care partners stay with them? Because I think this would be like a great help. Again, a personal experience was my stay in the hospital following my surgery. I had to stay 3 days in the hospital, and at the time, COVID rules were still in place, so nobody could stay with me. And I think that was the most isolating, most lonely time of my entire life, having to spend these 3 nights alone in the hospital. And I remember one night, I called the nurse to help me with something, and I just basically broke down in her arms and scared her because she thought I was having some kind of emergency, but I was just being overwhelmed, being alone after having such life-changing surgery. So I can imagine this be the same for everybody who is going through treatment through a clinical trial would feel the same. So these barriers we talked about are at the patient levels. On the next slide, we can see some of these barriers that are based from provider level or clinical level barriers. Again, like Dr. Strickler mentioned, providers sometimes have very limited awareness of what's going on, of trials that are available, and patients are left to find out by themselves if they qualify for any trial or if there's a trial that they can participate in. Another barrier is the time constraint. We all know that doctor appointments are like 10 to 15 minutes at the most. And I believe some clinicians, some providers would think that, okay, I don't have time to see the patient and to verify their eligibility to the trial, to explain everything to them, and to answer all their questions. So they just, that's a time constraint, that's a big barrier. Other times, even if they do suggest the clinical trials, they may face non-cooperation from their colleagues, particularly if there's another doctor on the patient team that the patient has known maybe for a longer time, and for some reason they listen to them more. At the clinical level, a big barrier to clinical trial participation is the restrictive eligibility criteria, which we know is there for a reason, but it still is a big barrier to the participation. Also, the complex clinical design and the complex strategy of implementation strategies, from recruitment, retention of patients, getting all the regulatory approvals, the ethical reviews, the operational logistics of coordinating across multiple sites, managing the data privacy, and which all of course amounts to cost millions of dollars for probably a single phase. These are only a few barriers I am mentioning, but these are all things we need to talk about and we need to address. Next slide, please. And this is, I told you all of this to tell you that this is where advocacy comes in, and this is why I do the work I do, because I believe it is very important to raise awareness, to educate patients, to educate providers about biomarker testing, about clinical participating in clinical trials. It is our duty also to promote the access to this information and to these treatments and to this testing to people across all populations, from whatever backgrounds, from whatever social economical status. And we do that by talking and by sharing our personal stories and the impact of these new technologies that have had on our treatment, because advocacy is strongest when it's personal. And this is why people in the advocacy space are willing to basically relive their trauma on a daily basis, because they believe very strongly in the power of advocacy to advance care for everybody. If we go to the next slide, please. And this is why I joined the Fight CRC Research Advocacy Training and Support Team, because I want to help other people. I want to make sure patients have a seat at the table, that they are included in all the decisions that are made on their behalf. I want to make sure that everybody gets the treatment they deserve. And I do believe that with working with patients and advocates, we can shape the progress of cancer care and make it accessible to everybody, and we can cultivate and improve the public trust in science and in medicine. And thank you for listening.
Thank you, Daad, for sharing your personal experience. And I want to tell you, there were people that joined the webinar today, particularly to be able to hear from
thank you.
A patient directly. So
thank you. That's great to hear.
Absolutely. I'm just going to close this off, or start to close this out with a couple of key takeaways. First, understanding HER2 and asking about biomarker testing early. This can really make a difference in treatment decisions. Secondly, if your cancer is HER2 positive, knowing that there are targeted therapies and clinical trials that are designed specifically for you, and that these options are expanding quickly, as we've heard, and it's worth talking about with your care team. And finally, as we've heard from Daad, your voice truly matters. By getting involved in advocacy, whether it's through programs like Fight CRC's RATS or our ambassador programs, or by simply sharing your story so that others can hear that there is shared experience, there is something, this isn't, they aren't alone. You are really helping drive real progress in research and in care. So I mentioned in the chat that I saw that questions were kind of coming in actively throughout the webinar. So thank you for doing so. Thank you to our panelists for answering the questions. We will, in the interest of time, kind of mull over the Q&A. We will compile a full recap of all of the questions and answers that were asked in the chat, as well as asked to each of our specific panelists, so that it is available to each of you to look at later. It will be available soon on fightcrc.org. And as I mentioned, it'll also be featured in November's clinical trials newsletter. So just wanted to kind of wrap us up further in how to get involved with Fight CRC. You can see what's here and available on our screen. But most importantly, we are excited to share that there will be 2 paid focus group opportunities for survivors of metastatic colorectal cancer, not limited to HER2, just if you are a survivor, that will be taking place over the next month. So this is a great opportunity and way for you to share your story and help shape future educational efforts. So we will follow up by email with more details on how to participate if you are interested. Additionally, we will send out the meeting recording and a couple of questions to get some feedback from you. And if you have any additional questions that you weren't able to ask just yet that still kind of came to mind, feel free to send them our way. And we'd like to extend a heartfelt thank you to Pfizer for their support of this educational effort. All content shared today was independently developed by Fight CRC and our panelist speakers, reflecting our ongoing commitment to empower patients and caregivers through trustworthy, evidence-based information. And as we wrap, we want to leave you with a reminder of why we do this work. At Fight CRC, our mission is what drives every effort like today's webinar. We fight to cure colorectal cancer and serve as relentless champions of hope for all affected. We do this through informed patient support, impactful policy change, and breakthrough research endeavors. This concludes today's webinar on understanding HER2, what patients need to know. On behalf of Fight CRC, thank you to our speakers, Dr. Benson, Dr. Strickler, and Daad, and to each of you for joining us for the launch of our clinical trials webinar series. If you'd like to rewatch this session or share it with others, the recording again will be available soon on Fight CRC, or you'll receive it directly in your inboxes. So until next